So as we look at photosynthesis, you're going to be pleasantly surprised because photosynthesis is going to look very much like the electron transport chain, at least a portion of photosynthesis. And photosynthesis, rather than being one process, is actually two processes, the light reaction and the dark reaction, and we'll give them different names. But that's essentially what's going to be happening. And in plants, where this happens is in our plastid called the chloroplast. So like the mitochondria, how many membranes does a chloroplast have? It has two membranes. It's got its outer membrane, it's got an inner membrane space, and the inner membrane's all folded up. Sounds very similar. But the chloroplasts fold their membrane into these stacks of coins called thylakoids. And you can see a lot of these stacks, it's like stacked pennies, the thylakoid. And when you take one whole stack together and consider it, it's referred to as the granum. And then you've got these little interconnecting portions of membrane, and that's called the stroma. But it's within these inner membranes, the thylakoid membranes, that's where we're going to have many of these proteins that are important for photosynthesis. So as we look globally at photosynthesis, we take 3 CO2 molecules, water, energy we obtain from light, and we're going to put those carbons together, assemble them into a 3-carbon molecule. And so we're going to capture the light energy. We're going to convert those photons of light into chemical energy, so that's referred to as a transduction of the energy. And then we're going to have something that's called carbon assimilation. That's the putting together of these individual carbons from CO2, combining them together into a single carbon molecule. So in our illustration on the right, we can see our light energy coming in. There are complexes of proteins on the inner membrane. You can see the electrons flowing. Does that not look like electron transport chain? But in this sense, what's happening with our electrons is we're coming along and we're producing NADPH. We're capturing the electrons that were generated from the energy of the photons of light. That energy then is going to come down and be plugged into this cycle called the Calvin cycle. And the Calvin cycle is where we're going to use the energy from sunlight. We're going to bring in CO2 and we're going to assimilate that together and produce starch. And once we transport some of this out into the cytoplasm, we're going to make sucrose. So this is the big overview, the global overview of what's going on. But remember, with our electron transport, we also pumped protons in our oxidative respiration. Well, we're doing the same thing also. Look at all the protons in our inner membrane space, and that's going to flow through. What in the world does that look like? ATP synthase. It is, because you're going to use that to produce ATP, and that ATP is also going to get plugged into the Calvin cycle. So from the sunlight, we pump protons. We've got our NADPH, electron transporter, but it's mostly those proton pumps that produce the ATP that we're going to plug in to use to assimilate the carbon into a larger molecule. We're bringing in energy. We're going from more disorganized to more organized. So what kind of reaction would this be? What kind of chemical reaction? We're having to bring in energy, going from disorder to order, endergonic, endothermic. What's the delta G going to look like? Positive delta G. And we're overcoming that by the introduction of our energy. Now again, we're not going to belabor this point. We've already talked about it when we introduced the organelles, but this endosymbiotic theory about how we had this, notice this is not a karyotic. They call it proto-eukaryotic because we got a nucleus, but we don't have much of anything else. And for our plants, it was said that, okay, well, we've got this aerobic purple bacteria that lived all by itself millions of years ago. And it said, you know what, I'm going to come in and I'm going to set up shop in this particular cell that's real good at organizing its genetic material and reproducing itself. And then the theory, the hypothesis also says at some point some green cyanobacteria, real good at photosynthesis, it comes in and it's going to set up shop. And so the purple became our mitochondria, and the green, the cyanobacteria, became our chloroplasts. That's the hypothesis of how possibly these two different organelles, the chloroplasts and the mitochondria, obtained those four membranes and are able to function in animals with just mitochondria and plants that have both. Because remember, mitochondria are in both plants and animal cells. It's not that chloroplasts are in one and mitochondria are in the other. So how do these photopigments work? They work by essentially absorbing wavelengths of light. And so if we look at the visible spectrum, we're going to see that a lot of that visible spectrum is what actually gets to the surface of our planet. It's a good thing that a lot of the UVs and others are reflected out in space because of our atmosphere. But we're basically working with wavelengths of light in the neighborhood of 380 to 760 nanometers. That's the wavelength. And when we look at chlorophyll, and we have a lot of different ones, chlorophyll A and B, we have the carotenes, and we've got the phycocyanins, the blues, and the phycoerythrins, that's red. And when you look at their wavelength and absorption peaks, you're going to see those all fall within the visible spectrum of light. And these are largely going to be responsible because of the colors that are reflected with the different colors we have in our foliage and our plants. So we're going to pretty much stick with chlorophyll. Chlorophyll A and B, these are going to be the ones that absorb blue and red, but they reflect the green. And then we also have these other accessories. The carotenoids absorb blue, but they are going to reflect others, so they're going to appear orange or yellow. The phycobilins absorb blue, green, and yellow, so they're going to appear red. Now, when are you ever going to see reflected light from the carotenoids and the phycobilins? No, not the flowers. They have pigments, and those may be mostly the leaves. In the fall. Are they just produced in the fall? Why do you see them then just in the fall? The chemical change of adenine, and that chemical changes, the chlorophylls aren't being produced, they go away. So the chlorophylls predominate when the leaf and the plant is living, and once that leaf begins to die, then the chlorophyll's not being produced, and so now when this goes away, these are still present and you see those different colors. And that's why they refer to these as accessory and these as the primary pigments. Does that make sense? Because that's how we can tell the leaves are dead, because they change color.
There's a few chlorophyll that makes the plant look red, like a red purplish color.
And do you have an example of those plants?
I couldn't
Yeah.
off the top of my head, but their leaves are like the whole plant will just
Oh,
happen
wow.
to be like a purplish color.
Oh, there is one. I can't think of it. It's
They're
kind of
also-
serrated leaf. You know what I'm talking about? It looks kind of frilly. Oh, shoot.
They're called also accessory pigments.
Yeah.
Because it's not that common.
You know the plant I'm talking about, too. I can see it. Can't get it. All right. So chloroplasts, where we have the chlorophyll, these are going to be embedded in those membranes again. The thylakoid membranes, the inner membranes of our chloroplast. Thylakoid stacked as the granum, with the stroma interconnecting all the grana together into one inner functional unit that we have inside our chloroplasts. So photosynthesis, we said, was this big box. Well, now we break it into the two boxes. The light reaction and carbon fixation, the Calvin cycle reaction. A lot of people refer to this as the dark reaction. The reason that botanists don't particularly care for that, and I sort of hate to say it, is this doesn't just happen at night. So it's not just in the dark. Okay, it can't happen anytime. So the light reaction, this is where we're capturing the energy from the sun. And we're going to convert that light energy into our chemical energy in the form of ATP and the NADPH. That's the one in plants. That energy then is going to drive the carbon assimilation as we begin the process of forming our carbohydrates. So another way that you can describe this, light reactions, light independent reactions, much more so than just dark. But this is also called the Calvin. The Calvin cycle is a part of the light independent.
Desert plants use that form of light independent reaction. Just simply because if they were to do it during the day, they would burn up in the night.
You know another interesting thing about humans and animals? Your skin is there to largely protect you. Well, your skin's going to be exposed to ultraviolet radiation, which can cause damage to the DNA, which can lead to mutations and eventually cancer. Do you know one mechanism through which your skin cells try to protect themselves from ultraviolet radiation other than the pigmentation? Your skin cells only divide at night. Your skin cells don't undergo mitosis during the daytime. Or at least they slow down. So when you're replicating DNA, it's away from the ultraviolet light. I'm like, well, that's pretty sharp. Okay, so here's our illustration. Light independent reactions. Light dependent, sorry. We're bringing in water, giving off oxygen, so we're really glad that plants can do this. There's our electrons. There's our substrate level ATP. This is happening in the thylakoids, the stacks. But in the stroma, the interconnecting strands, this is where we have the light independent reactions, where we're bringing in the CO2, also giving off water instead of picking it up. And we make our first carbohydrate, that 3-carbon sugar. And then we've recovered the electrons, we've recovered the energy, and we recycle the NADP and the ADP back into this, and it just keeps going. And we like to eat those. So we help, I guess, no, we don't really help the plants because we eat the plants. So we're going to start with the light reaction first. Now, this is an interesting illustration, but we're going to see our graph. Do you remember that graphic that we had of our electron transport chain that started with our electron and it just kind of passed the electron and went down and down to oxygen? This is sort of the same thing. They're building it up. See, it's higher around the outside. And our photon of light, when it activates the photopigment and energizes it, it's going to send this to a higher and higher energy state until at the very end we donate that electron to our electron acceptor and charge up the NADPH. So this is almost like it's rolling downhill, but from an energy standpoint, we're going uphill. And the reason we can go uphill, endergonic reaction, is because of the injection of energy at that point. So here is that kind of graphic again. So here we start with our water. We're passing it through our photosystems. And here we have a photopigment that zapped with a photon of light energizes it. Now this is also, they call this our energy, activation energy, but that would also be like delta G. Delta G negative goes down, but a delta G positive, that's an endergonic, but that's because we're putting in the energy from our photon of light. Here we come through our system. The next photosystem, we've got our like a cytochrome B6. So we're passing electrons along, the energy along, but there's our proton pumps, just like we saw in our pathway. We get to another photopigment, charged it up with energy, boom, higher energy, until finally we get to a point where we can add the electrons to NADP, and we've captured the energy from these photons of light. But again, in addition to capturing the electrons, we've also pumped out these hydrogens that we're going to use to make more ATP. But do you see the similarity? The electron transport function here, in addition to the photon, it's the photons of light that are really the only difference that we have from our electron transport chain in our mitochondria. Very, very similar processes. So it's kind of interesting how this is drawn upside down compared to what we have with our animals, and maybe that's a way to distinguish. But again, here's more of a cartoony look. Our photons of light coming into our photosystems. There's more photons of light. You can see our electron transport, our proton pumps, and then our ATP synthase. And they call it CF in plants. I don't know what the C stands for other than chlorophyll or something. But there's the F0 and the F1. Protons flowing through, generating the production of ATP. So if you learn this portion, this light-dependent portion, and you learn the electron transport chain, there's very little that's going to be different between them. That difference is going to be the injection of energy and the photons of light, more so than the bringing in of electrons from those cofactors. Do you see the similarities that we have? All right, so that was our light-dependent capturing of electrons, pumping of the protons that we use to produce our ATP, oxidative phosphorylation of ATP. And again, we've already seen that through our mitochondria. Now, as we get into the light-independent reactions, here's the Calvin cycle, carbon assimilation. So here are our 3 carbon dioxide molecules that we're going to bring in. We have these 3 molecules of rubulose bisphosphate, Rubisco. And these 3 Rubisco molecules, notice 1, 2, 3, 4, 5 carbons. So we have 15 carbons here altogether. We're going to combine them with our 3 CO2s, 18 carbons we're going to have to play with. And so as we bring in the CO2s, we use our Rubisco, 3 Rubisco molecules, we end up with 6 3-carbon molecules. And with these 6 3-carbon molecules, here comes our 6 ATP. We're going to add the phosphates to these. We're bringing in 6 electrons. With the phosphates and the electrons, we're going to end up with 6, what is the G? I want to say glyceraldehyde 3-phosphate, but I know it's not glyceraldehyde 3-phosphate. These 6 3-carbon molecules that are going to go on then to be used to build up our starches. Now here we've got 6 3-carbon molecules, one of which exits the pathway. So we lost 3, we've got our 15 again. That goes through enzymatic processing. We're losing some phosphates, adding some phosphates, changing things around. So we go from 5 3-carbons to 3 5-carbons. We get back to Rubisco and we start the process again. So do you see the cyclical nature? We bring in 3 CO2s, we give off 3 that are assimilated together. So once again, we've got our phases, the carbon fixation, bringing in the CO2. This process we refer to as the reduction of the CO2 into PGA, and then the regeneration of Rubisco as we start the process again. So just before, when we talked about the TCA cycle, remember oxaloacetate was that acceptor that combined with acetyl-CoA to form citrate. Now we're looking at that same sort of process, but we're looking at rubisco along with the carbons to capture the CO2 and assimilate them into those 6 3-carbon molecules that we're going to process further on. And then the other part of this whole process is the understanding of bringing in the 3 carbons with CO2 and eliminating one of those 3-carbon products, the G3P, out to be formed into starch, into sucrose, as we repeat the cycle and reproduce that ribulose bisphosphate. Rubisco is the enzyme. Ribulose bisphosphate is the actual 5-carbon molecule. So ribulose bisphosphate, CO2, enzyme rubisco, we have those PGA molecules, and the PGA then being a 3-carbon sugar. So that's the process there in the beginning. When we bring in our carbon dioxide molecules, we generate our PGA, and by the time we get to the end of that middle portion, there's our glyceraldehyde 3-phosphates. That's what's going to leave, one of those is going to leave this whole process to go on to form the larger sugars. But understand, what are we having to input here? Energy. So we're talking about an endergonic reaction. Where do the electrons come from and where does the ATP come from? The sun and the light-dependent portion of the reaction. Without the sun, without the electrons, without the oxidative phosphorylation building up the ATP, we would never be able to put these together. We would never be able to generate that. So, here in the assimilation, the 3 ribulose bisphosphates, the 3 CO2s, there's that enzyme rubisco. We get our 6 PGA, phosphoglycerate. There's the ATP, the NADA. Again, energy coming from the sun. One of these pops off to go to make our sugars. There's our 15 carbons that are left. Our 15 carbons are recycled into the ribulose bisphosphate. We've got these enzymes along the way using energy of ATP. Again, where did that come from? The sun. The light-dependent reaction. So 15 carbons rearranged into 3 5-carbons, and we start the process again. So that's why I like to think of the 3 CO2 coming in, exit, all tied together. Any questions?
Does this happen in each layer of the thylakoid? Or?
The thylakoids do the light-dependent, and the stroma does the light-independent. So how is the Calvin cycle, how's it regulated? Well, if you don't have sunlight, it's not going to go anywhere. If you have more of the enzymes, then you're going to have more of the carbon assimilation. If you have some of these light-dependent byproducts around, these are going to be activators of those enzymes. So some of the products then of the light-dependent reaction, some of those intermediate steps, we can say they are allosteric activators of the enzymes in the light-independent reactions. You see how that works? So when you have a lot of these byproducts, meaning there's plenty of sunlight, you're producing and capturing a lot of energy, that signals these enzymes, hey, get busy and get this stuff processed because we're sending more your way. And that's why understanding how enzymes work, understanding what allosteric regulation really means, because we're applying it in situations like we do in photosynthesis. So we start the whole process of forming our sugars with glyceraldehyde 3-phosphate. And then we also have the dihydroxyacetone phosphate, and these kind of go back and forth. They're reversible reactions with a single enzyme. But if we're in the stroma, you were sort of asking about the compartmentalization. So where here in the stroma, where we produce this glyceraldehyde 3-phosphate, we can keep this around in the stroma, and we can, with these different enzymatic steps, we can form fructose 6-phosphate, glucose 6-phosphate. Sounds a little bit like the reverse of glycolysis, but we can go through these enzymatic steps. ADP glucose, assemble this phosphated glucose, boom, we can assemble that into starch. And so starch is going to be produced in the stroma of the plant. But we can also transport this glyceraldehyde 3-phosphate out into the cytoplasm. Fructose 6-phosphate, glucose 6, UDP glucose. Combine it with a phosphated fructose. There we have sucrose. Where do we know sucrose from? Table sugar. Sucrose is table sugar. And sucrose production happens in the cytoplasm, whereas starch, that's going to happen in the stroma of the chloroplast. So again, we've got a lot of transporters bringing in and producing these enzymes, transporting them in and out, transporting in and out the glucose and the sucrose. So there's a ton of traffic, again, happening in and out of the chloroplast with a lot of these intermediates, a lot of these reactants that we saw that transport happening in and out of the mitochondria as well. No. Mm-mm. No, the only enzyme that is extremely important to remember for photosynthesis is rubisco. Rubisco, it has its own name. It's going to be the enzyme responsible for the carbon assimilation of the CO2s. So that's really the only enzyme. Did we even mention the name of an enzyme in glycolysis and TCA? No. So that's really the only enzyme that... But rubisco has such a cool name, so, you know. Sounds like someone's name, rubisco. I don't actually think nobody's named that. Now, this is an interesting interaction and crosstalk between three separate organelles. That's kind of what makes it very interesting to me. Between a mitochondria, a peroxisome, and a chloroplast. So when we look at this pathway called the glycolate pathway, this is going to be a process by which we recover 3 of the 4 carbon atoms that exits the Calvin cycle. But in addition to that, we're going to make sure that this phosphoglycolate, this intermediate that's being produced, is not going to build up to toxic levels inside of the cell and inside of our organelles. So when we look at our Calvin cycle, here comes that phosphoglycolate, and if that builds up to too high a concentration, then it's going to drive a reaction through this enzyme to convert that into glycolate that gets transported into the peroxisome. The peroxisome then is going to have enzymes and materials that can convert the glycolate into glycine. What's glycine? What's glycine? We know what glycine is. Glycine is our most fundamental amino acid. Only a hydrogen as a side chain. Yeah, kind of bringing that back now. We're going to use everything we've learned just in different bits and pieces now. So here we have glycine that in the mitochondria, enzyme's going to react on it to produce serine, another amino acid. Yeah. So we just changed it. Now we take serine, ship it back into the peroxisome. We convert it into glycerate in the peroxisome that gets transported back into our chloroplast as 3-phosphoglycerate. That gets plugged back into the Calvin cycle. So do you see, it's like, okay, we don't want the phosphoglycolate to build up, so we're just going to have it spend a lot of time going around and about in a circle before we plug it back in, just to keep this at lower concentration. But we're not necessarily taking away from photosynthesis in the Calvin cycle, because even if we ship it out, we're bringing it back. But notice, to be able to do this, we're expending some energy. We're getting rid of some electrons. Yeah, we do kind of capture some here in the mitochondria, but this is going to be at the expense of using energy to accomplish this shuttling around to prevent that toxicity and that buildup. So that's just an interesting little regulatory pathway, sort of as an emergency backup plan. I think if you just look at this simplistic flow chart and you look at CO2 in, phosphoglycerate out, phosphoglyceraldehyde, out, the energy coming in from the sunlight, and we could put sun, photon of light coming in, that might help too. But this is green because of photosynthesis and light. This is not green because it's the light independent part of the reaction. So that's the summary, the synopsis of photosynthesis. And again, it is so very similar to what we saw with our electron transport chain. And it is in some ways similar to the TCA cycle. The Calvin cycle is. TCA, you're liberating CO2. Calvin cycle, you're assimilating CO2. So I think it's kind of interesting and cool to put these two chapters so close together just because of how similar they are. And really, when you think of it, the mitochondria and chloroplasts are similar in a lot of different ways, structurally, not just functionally.